proteins they make. So that developed proteomics. That became a hot topic. Okay? And so what I'd like to suggest is what we are doing today is that we are exploring proteomics. We're looking at which proteins can a microbe make. And specifically, we're going to look at which enzymes can a microbe make. And it turns out that the set of enzymes that a microbe makes, it's like a fingerprint or like a zebra code. Every organism makes a unique set of enzymes. So we can use that set of enzymes to help us identify unknown bacteria. And that's really what you guys are going to be doing after the break. You're going to be doing the last chapter in your lab manual, which is the unknown chapter. You're going to be given 2 unknown bacteria, and you're going to use all the techniques, your gram staining, your streaking for isolated colonies. You're going to be using the metabolic test that we teach you in the next few labs to identify these 2 unknown bacteria down to the species level. And you guys, that's what professional microbiologists do. You will have the skills to go out in the real world and do all kinds of good, identifying unknown microbes, identifying pathogens that are growing in your patients. Okay, so this is really powerful stuff. And like I said, it's really fun. Okay, so that's the big picture here. We're trying to figure out which enzymes our different microbes can produce. And what we're going to do to help us identify these enzymes, we're going to incubate our bacteria with different types of media, different types of substrates, and we'll look for production of end products, or we'll look for production of unique intermediates to help us identify indirectly the enzymes our microbes are making. Okay, so we're going to start then in chapter 16. This is often referred to as Metabolic Test Part 1. We're going to introduce you to some really fun metabolic tests. And the 3 metabolic tests we're going to introduce you to today are the PR sugar fermentation tests, thioglycolate test for anaerobic growth. And then, although we're not going to run the test, we're going to describe an important test that's used in diagnostic bacteriology called the oxidase test. Okay, so these are the 3 tests that we're going to describe today. And they're all described, you guys, if you haven't had a chance, they're all described in your lab manual. So as always, you guys, make sure that you're reading the introduction and reading the text of the lab. Okay, so what we'll first do is talk about the Phenol Red sugar fermentation tests. And what I'm going to do is refer you to page 142. There's a little table there that will help guide us to know which sugar fermentation media we'll use with our different bacteria. So that's just so we have a little place of reference. Now, the theory behind these PR fermentation tests is that we have a media that contains a single sugar. Okay, and this is a so-called PR sugar fermentation broth or media. And it's really important that we know the components, what goes into this PR sugar fermentation media. Okay, so first of all, the PR stands for a pH indicator. Any guesses, you guys, on what PR stands for?
Phenol Red.
Phenol Red, dynamite. Excellent, you guys. So, the media, when it's fresh, sterile, before we inoculate it, it appears red, and it's because of the phenol red pH indicator present. Okay, so that's what the PR stands for. Now, in the media, only a single sugar will be present. So today, we're going to use 3 different types of sugar fermentation media. We're going to use PR glucose media. So which sugar is present in PR glucose? Glucose, right? Okay, and just so you know, and I'm not sure historically, you guys, why this is. Usually in medical settings, they refer to glucose as dextrose. So these have been labeled as PR glucose / dextrose, and just recognize it's the same thing. Okay. Another type of PR fermentation media you'll inoculate is PR lactose. Which sugar is present in PR lactose? Lactose, good. You got the hang of it, huh? Okay, and the last one is PR sucrose. Okay, and so which sugar is present in PR sucrose?
Sucrose.
Sucrose, good. Okay, now just quick chemical chapter 2 review, you guys. Glucose we know is a monosaccharide. And it turns out that if organisms can ferment sugars, usually the first one they're going to ferment is glucose. Okay? Now, do remember, you guys, that lactose is a disaccharide. So what are the monosaccharides that make up lactose? Glucose and galactose, right. And remember, if a microbe is going to use lactose as a source of carbon and energy, it has to have an additional enzyme. It has to hydrolyze the glycosidic bond between the glucose and galactose. So which enzymes do bacteria make that will hydrolyze that glycosidic bond? Think about the lactoperon we talked about today. Think about the LAC-Z gene. The LAC-Z gene encoded which enzyme? We called it bacterial lactase, but what's the real name? Beta-galactosidase. Dynamite, you guys. So if an organism can ferment lactose, it has to produce one more enzyme. It has to produce beta-galactosidase. You know, one more enzyme compared to just glucose fermentation. Right? Okay, good. And do you guys remember the structure of sucrose? Is it a monosaccharide, a disaccharide? It's a disaccharide. It's often called table sugar. Do you know the components of sucrose?
Glucose.
Good. Excellent, you guys. Fructose and glucose. And so which enzyme do you think would hydrolyze a glycosidic bond between the fructose and glucose? Sucrase. Excellent. Dynamite. Excellent, you guys. Okay, so remember that if your microbes can ferment sucrose, they have to make this special enzyme sucrase, right, to break that glycosidic bond. Dynamite, you guys. Excellent. Okay. So we said in the PR sugar fermentation media, we have Phenol Red, we have a single sugar. Okay. Oh, and I forgot to tell you this, guy. Can you distinguish between PR glucose, PR lactose, and PR sucrose based on color? Yeah. Well, we should. It should all appear the same. I know that the glucose one's looking a little bit more orange. So it's very important, you guys, that before you come up to grab your tubes, make sure you make your tape labels. And you're going to work in teams of 4. And each team of 4 is going to have a total of 3 PR glucose tubes, 3 PR lactose tubes, 3 PR sucrose tubes. And you want to make sure you make your tape labels before you come up, because if you grab the tubes and take them back to your bench, you're going to be sitting in the corner here, Francis. You want to make sure that you put the tape labels on the appropriate tube. Now, if this happens, you guys, and I'm notorious for this, if you're up here getting your tubes and you're very organized, and I come up and I start going, you know, bada 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 bada, and you just picked up this tube and you're like, oh, sugar, I can't remember what tube this is. Don't worry about it. Just loosen the cap and put it in the kill area, okay? Because you don't want to use it and mistakenly put the wrong label on it. And what's really important, you guys, you don't want to put it back for somebody else to pick up if you made a mistake. No big deal. Just loosen the cap, put it in the kill area, okay? If you forget. All right. So we talked about the Phenol Red, we talked about the sugar. Now, in addition, in the media, there will be an alternate carbon and energy source, because we're going to discover some of our microbes can't use these sugars, and we need for them to grow. You know, this test won't work unless the microbes are growing. So we have to provide them with an alternate carbon and energy source, and that will be proteins and amino acids in the media. Okay, so if your microbes can't use the sugars, what they'll do instead is they'll tear apart the proteins down into the individual amino acids. And you guys know this is kind of your generic amino acid. There's the carboxyl group, there's the R group, the central carbon, and then here's your amino group. So if your microbes have to use the amino acids as a source of carbon and energy, they tear apart the amino acids, and the amino group gets released as ammonia. It's called deamination. That amino group is released as ammonia. And you all know from chemistry that ammonia is a weak base, right? It can bind hydrogen ions in solution to form what? Ammonium, right? Okay, so let's think about this then. If your microbes can't use the sugars, they have to use the amino acids instead, they release ammonia, which binds up free hydrogen ions, what's going to happen to the pH of your media? What's going to happen to the pH? It's going to go up, right? Okay, so that's going to be important. As long as we're talking about pH, let's think about what happens if sugars are fermented. Okay, if your organism can ferment the sugar, what is one of the most common fermentation end products? One of the most common fermentation end products are acids. Okay, so if your organism can ferment the sugar that's present, what's going to happen to the pH?
Go down.
It's going to go down, right? So let's think about this. How will we know on Tuesday when we pull our tubes out of the incubator, how will we know whether the sugar was fermented? Well, if the sugar was fermented, the pH will decrease, and what color will the phenol red turn?
Yellow.
Yellow, dynamite, you guys. So yellow indicates that acids were produced from fermentation. So it's a positive sugar fermentation test. Okay? Now, in addition, during fermentation, some microbes have additional enzymes that permit them to make, so I'll put plus or minus, some do, some don't, gases. And the gases are usually either molecular hydrogen and or carbon dioxide. And that too is important. It's important for us to detect whether gas is produced. So the last component in the phenol red sugar fermentation tubes is this little inverted glass tube, and it's called a Durham tube. Okay, so that little inverted tube is called a Durham tube, and its function, Durham tube. Its function is to collect gas released during fermentation. Okay, so what you will do is you will record not only the color of the medium, is it yellow, indicating acid production, or is it deep fuchsia, indicating that we had a base produced, wheat base produced from deamination of amino acids. You'll always also look for a gas bubble. Okay? And again, you guys, if we have gas present, what would the gases be? Molecular hydrogen or CO2. Those are the most common gases produced during fermentation. Okay, so let me back up here, and I've forgotten which this belongs to. Okay, so let's go ahead and use our chart here to look at the three different results we could get. Okay, so here's our uninoculated tube. You're going to use dynamite aseptic technique today to transfer pure cultures that Val and Carmen made for us growing on plates, and we'll describe this. You'll inoculate your tubes. You'll have excellent labels on it. You want to put the name of the sugar, the date, the name of the microbe, and at least one person's name from your team, and the lab section. So those are our labels. We'll transfer our microbes. We'll make sure that the caps are just barely loosened so they can vent. Now let's presume we have a microbe that can carry out fermentation of the sugars with acid production. So when you pull your tubes, then on Tuesday you would expect to see the media is yellow, indicating an acidic pH, right? Now some microbes lack the extra enzymes to make gases, so we could see just acid production without any gas. And we've got a scoring system for this. So we score for acid, we score for pH, and then we score for gas. So if we have a tube that's all yellow with no gas bubble, we score at acid / no gas. And we even can abbreviate it even more. Sometimes we'll just put A / negative like that. So that means acid was produced. We had fermentation of the sugar, but no gas was produced. Now some of the microbes, as we said, they'll have extra enzymes. They'll actually make molecular hydrogen and or CO2. So we'll see yellow media indicating acid produced from fermentation of the sugars with gas. So we can score that as A for acid and plus for gas. Now, the microbes that lack the enzymes to carry out fermentation of the sugar, like we said, they'll attack the amino acids, they'll deaminate the amino acids, releasing ammonia. The ammonia binds the hydrogen ions. The pH increases, so the media will turn red, okay? And that is telling us we have an alkaline reaction. Okay, so what we do is instead of using A for alkaline, we use K. Okay, K for alkaline. And if we have, if the microbe can't ferment the sugar, there should be no gas. So if you have an alkaline reaction, it should always be gas negative. So that's how you're going to score your tests next week. So which tubes indicate sugar fermentation? This one and this one, right? This one is a negative result for sugar fermentation. The organism could ferment the sugar. Now, there is a problem, though. There's a problem. And that is, let's say that we inoculated our microbe and they could ferment the sugar, but we left the tubes in the incubator for, say, 7 days. Maybe we get sick next Tuesday, can't pull the tubes. Well, your microbes are going to use up all the sugars and they'll start to starve. So if you were them, what would you do if you were starving because you ate all the sugar? Is there another carbon and energy source in the media that you could use? The amino acids, right? So if you over-incubate your tubes, with an organism that can ferment the sugar, you'll get the yellow change, the yellow color change when the acid is produced. But if you leave the tubes in an incubator too long, the sugars are used up, and then your microbes will start attacking the amino acids. So after like 7 days, you'd come back and you'd have a red color, and you say, oh, my microbe didn't ferment the sugar. But would that be correct?
No.
No, that would be a false negative, right? And you just want to think of that if your tubes incubate. Usually we want to leave them for 48 hours. Okay, so once we get past 48 hours and we start getting negative results, we're a little bit, we're not sure. Is that a false negative or is that a real negative? The one way that you would know that it's a false negative is if you have the red color, but there's a gas bubble up there. Okay, because the gas bubble would only be produced during sugar fermentation. Okay, so that might make you think, oh, maybe I left these in here a little bit too long. Now, you can also get a false gas bubble if you take your PR tubes and if you turn them upside down like this. Okay, you can see right now I'm doing it. I'm introducing an air bubble. Okay, and that would give you a false. There it goes. Yeah. That would give you a false gas positive reading, and it's just an artifact. So make sure you guys don't turn your tubes upside down like that. Okay. So that would be another way where we get a false air bubble in there. Now, you guys know, okay, is today Thursday? Today is Thursday, right? Okay, so if we put your tubes in there today, so Thursday, Friday, Saturday, Sunday, Monday, Tuesday, 5 days, that's way too long to incubate these. So that's why it's so important that we put all of our Tuesday, Thursday tubes in that middle 37-degree incubator, because that incubator will be turned off Saturday. Okay, and we do want to give them 48 hours. This incubator over there, that's the Monday, Wednesday incubator, so that incubator will be turned off tomorrow. So it is kind of important that you put your tubes in the right incubator. Okay, so we'll turn the incubators off Saturday so they don't over-incubate. Okay, so if we take a look now at that table on page 142, okay, let's see who you're going to inoculate. Okay, so again, you're going to work in teams of 4. And you want to make your labels first. That's really important. Okay, and I'll just put the chart up here. So we have PR glucose, PR sucrose, PR sucrose, and we have PR lactose. And then I'll also add that thioglycolate because we're going to talk about it in just a few minutes. Okay. And then we've got your pure cultures. So one set of tubes you'll inoculate with alkalogenese faecalis. And Carmen and Val gave us pure cultures growing on plates. Okay, so the alkalogenese faecalis is A. faecalis on the bottom of the plate. So we have enough plates for 6 teams. Yeah, so we might need to combine teams, you guys. Unfortunately, we're losing some of our colleagues. So if there's only maybe 1 or 2 of you, maybe you could combine with another team, okay, because we only have enough plates for 6 teams. And then the next organism you're going to inoculate into a second set of the PR tubes is Bacillus subtilis, our close cousin of Bacillus anthracis. Okay, and see, Bacillus subtilis says B subtilis on the bottom of the plate. And then our good friend E. coli. E. coli will go into a third set. Okay, so each team of 4 will have 3 PR glucose tubes, 3 PR sucrose tubes, 3 PR lactose tubes. Okay, and each of you 3 microbes you'll inoculate into the 3 different sugar tubes. Okay, does that make sense? Okay. All right. Now, the next media that we have listed in that table, it says thioglycolate, and you'll see that there's an Na where Bacillus subtilis sits. That means we're not going to inoculate. We will not inoculate Bacillus subtilis in the thioglycolate. It's kind of pricey media, so we're going to limit the thioglycolates to just 2, to our Alkalogenes faecalis and our E. coli. Now, there is a special technique for inoculating these tubes. So let's take a look at them. So the thioglycolate media is up in the rack, and it's really important you don't shake it, you guys, and we'll explain why. Okay, so this might ring a bell from lecture when we were describing microbial growth in chapter 6, and we were talking about microbes' relationship to oxygen. And you might remember the diagram that showed the 4 different patterns of growth. Well, you now are going to run that experiment. Okay, so thioglycolate is a substance which reduces molecular oxygen and thus creates an anaerobic environment. Okay, so you'll see in the media that the bottom portion of the tube, it has a slight yellow color. That's the anaerobic portion of the tube where the thioglycolate has reduced the molecular oxygen. Now, in addition, there's an oxidation reduction indicator in the media, and it turns green when oxygen is present. So this makes sense. We've got a little aerobic zone up at the top where we have a little bit of molecular oxygen continually diffusing into the media. Okay, so this is our little aerobic zone there. The rule of thumb is you never use thioglycolate media if more than 1/3 of the tube is green. Okay, and usually what you just have to do is re-autoclave it. Okay, and that will return it to primarily anaerobic. So again, it's really careful, guys. It's really important that when you come up and get your media, don't jostle them, don't shake them. Okay, because we need to keep this portion anaerobic. When you inoculate the tubes, you can use your loops to inoculate them, but what you want to do using good aseptic technique is you're going to go straight down and straight back up. You don't want to jiggle your loop like I showed you because you don't want to introduce oxygen. Okay, so you're going to go straight down and straight back up. Okay, as always with these screw cap tubes, you want to tighten them and then just barely loosen them so they can vent. Good labels, very important, you guys. You're going to transfer all of your tubes into your well-labeled cans, and they'll go in the 37°C incubator, the middle one here. Okay, so the thioglycolates, you'll inoculate Alkalogenes faecalis into a thioglycolate, and in a separate thioglycolate, you'll inoculate E. coli. Okay, so those are the transfers you'll be performing today. Now, the last topic, you guys, and then we'll let you get to work, is we just want to describe the oxidase test. Now, the commercial oxidase test that we used to use, we were getting really inconsistent results, and with the budget cut back and everything, we eliminated it from our so-called wet lab. But the oxidase test is a real important test in diagnostic microbiology, so I wanted you guys to know about it. And there's a description of the oxidase test on page 139 up at the top. It describes the oxidase test. So the oxidase test is a test for a specific member of the electron transport chain. Okay, so it tests for the presence of a specific electron transport chain component, which we're calling cytochrome oxidase. Okay. Now, the reason this test is important is it's used to help with the initial differentiation of some gram-negative bacteria. Okay. So it's important when you're working with gram-negative bacteria? Gram-negative unknown bacteria? Okay, so we'll do one of our little flow charts. Okay, so again, we're going to use this when we're working with unknown gram-negative bacteria, and we're trying to figure out, what is the identity of this unknown gram-negative. So if we run the oxidase test, just information I'd like you to know for the lab practical is if we're oxidase negative, this is classic for that huge family, Anaerobacteriaceae. Are you guys all right? Oh, it's a little spider? Oh, don't hurt him. Oh, good.
It's still alive. It's still alive. I got it.
I know, I'm so silly. I'm so sorry.
Are you recording this? This would be great.
It'll kill us.
Okay, sorry. You guys are so funny. Oh my goodness. Okay, so the oxidase test, that big family of Anaerobacteriaceae, they're all oxidase negative. Okay, so, and this is a huge family of bacteria. It includes E. coli. Felix, thank you. That was very kind of you to protect folks. We've got E. coli, E. coli, Salmonella, important pathogen, Shigella, Yersinia pestis, which causes bubonic plague, Anaerobacter. These are just some examples. This is a huge family of gram-negative bacteria. And in fact, I would say the majority of the gram-negative bacteria we work with here in lab are Anaerobacteriaceae. So an important part of this family is they're all oxidase negative. Now this is misleading, you guys, because you might think, oh, they're oxidase negative, that means they don't have an electron transport chain. But is that true? What do you know about E. coli?
It can go off the
It can, wow, does it have electron transport chains, right? Because we know that E. coli is a facultative anaerobe. If oxygen is present, it carries out aerobic respiration using one kind of electron transport chain. If oxygen isn't present, but say nitrate is present, it can switch over to anaerobic respiration, and it has a different electron transport chain for anaerobic respiration. And then we know if there's no alternate electron acceptor for anaerobic respiration, it can switch to fermentation, mixed acid fermentation. So again, you guys, since this whole family is oxidase negative, does that mean we know with certainty that they lack electron transport chains? No. Okay, I mean, it might be they lack an electron transport chain, or it might be they simply don't have that specific component of the electron transport chain. So that's something important for us to remember. Okay. Now, in contrast to the big family Anaerobacteriaceae, another an opportunistic pathogen, a real important opportunistic pathogen that's going to cause real headaches in hospital settings, are the Pseudomonas. And we do work with Pseudomonas here in the lab, you guys, and some of you might get it as your unknown. So Pseudomonas and the Anaerobacteriaceae on a gram stain, they may look pretty similar, right? So you're like, how the heck do I, what's the initial step to distinguish them? So that big group, that big genus of Pseudomonas, they're oxidase positive. Okay, so that's one way to separate them. And another group of gram-negative bacteria that are important pathogens are the Neisseria. So Neisseria gonorrhea that causes gonorrhea, Neisseria meningitidis that causes bacterial meningitis. They are also gram-negative. They're little diplococci. Okay, so it's hopefully you wouldn't confuse them with, say, E. coli, but they too are going to be oxidase positive. Right? So even though we're not going to run the test, you guys, I would, on the lab practical, I would like you to know that this big group of gram-negative bacteria, the Anaerobacteriaceae, they're all oxidase negative. And then I would like you to know these two examples of potential gram-negative bacterial pathogens that are oxidase positive. Okay? All right. So with that, you guys, we'll let you all get to work. So you might just first want to organize and see if you can combine teams. Okay, so ideally you guys would like to have 6 teams total because we only have 6 plates. So each group will need a total of 9 PR tubes and 2 thioglycolate tubes. And then each team, if you all would share one stock culture plate of the E. coli, the Acarigines faecalis, and Bacillus